Welcome to Lesson 2 of Module 6, Genetic Change. In this video, we're going to be looking at the genetics of mutations. Welcome to Lesson 2 of Module 6, Genetic Change. In this video, we're going to be looking at the genetics of mutation, and we're going to be looking at how genetic variation occurs and the effect of these mutations on populations. So in this video, when we talk about polyploidy, it means we have more than two paired homologous sets of chromosomes. Then we have monoploidy, an individual that contains 1/2 the normal number of chromosomes. And then we have aneuploidy, where we have a chromosomal mutation in which there is one or more extra chromosomes or one or more fewer chromosomes. When we have sexual reproduction, there are processes that occur in sexual reproduction that increase genetic variation. So in gamete formation, so during meiosis, we see this happening. And then further on to fertilization, we see an increase in gene recombination, and therefore we see a whole lot of new gene combinations being produced. This can be, and this variation that we see, can be as a result of recombination of genetic material, and then it can be an increased number of alleles for a particular gene being present in that population. So when during meiosis and the formation of gametes, we can see crossing over and random segregation happening. And therefore, we see genetic recombination of paternal and maternal genes within the gamete. When two different gametes unite during sexual reproduction, so this is the fertilization process, there's a further mixing of genetic material, and therefore we have offspring that have many new combinations of genes. Chromosomal mutations may arise due to errors in crossing over or non-disjunctions of chromosomes, which we looked at in the previous video, or the chromatids during anaphase 1 or 2. When we talk about chromosomal errors, this is when crossing over goes wrong, and this can cause DNA to be exchanged during the synapsis, or we can see a chromosome break, which can cause duplications and or deletions after anaphase, and then chromosomal errors can also occur due to exposure to mutagens during meiosis. Non-disjunction, which we looked at previously, is where the homologous pairs of chromosomes do not separate as they should, and therefore we get an abnormal distribution of chromosomes in the daughter cells from meiosis. Here we have another image, different to the one we saw in the end of the last video. We can see the gametes being produced here with a different number of chromosomes than what should be in there. So by being able to calculate these changes in genetic variation, we then can use our population genetics that we've also used in Module 5, and we can see changes in allele frequencies. So natural selection, a process which we're very familiar with, does cause changes in allele frequency, and this is due to choice. And then genetic drift is where we see changes in allele frequency due to chance. When we talk about gene flow, we're talking about new individuals entering a population or existing individuals leaving. So these are called immigration or emigration. And a fixed allele is an allele that becomes the only remaining allele in that population, and therefore it is outcompeted all of the other alleles. So Mendel, he studied population genetics, and he knew that each parent donated one allele, and that some alleles are dominant, others are recessive, and they can be expressed in single or two copies. And only recessive genes are expressed when they're not paired with a dominant gene. And then Darwin, he obviously figured that natural selection drives evolution, and then if a gene is advantageous, it's going to be passed on to the next generation. So there are 5 factors which can change allele frequency in a population. So we can see selective pressure, so different changes in the environment selecting different alleles. There's sexual selection, and this is due to mating not causing these different alleles to be happening in the population. Mutation is where we see new alleles arising during meiosis. Genetic drift, so this is where we see our random chance. So this might be because of a natural disaster, and this is where we see the bottleneck effect, or we can see when a population becomes geographically isolated from the original population, we can see the founder effect. And then we have our gene flow, and it's the changes in allele frequency due to the mixing of new individuals in the populations, therefore introducing different alleles. So here we have just an image that goes through all of those different ways which we can introduce variation into the population. And here's the founder effect and the bottleneck effect. So due to these critical events, we can either see the number of characteristics being lessened, or we can see them being isolated from the population and therefore having their own alleles in that population. That concludes lesson 2. Make sure you tune in for lesson 3.